Hey, welcome to this video on cell division. Here in this video, we're going to start to understand how and why cells divide, and we're going to look at similarities and differences among different types of cell division. So let's get into it. All living systems on the planet have to be able to reproduce, and since all living systems are made out of cells, cells have to be able to reproduce. If you start with one cell, you're going to wind up with more than one cell at the end. And so cell division is the way in which cells reproduce. It is the reproductive property of life writ large. If we think about a particular cell and it's living its life, we can think of its life as a cycle, what we call the cell cycle. And so what we see here is a diagram showing the major parts of the cell cycle. Cell division is this tiny little slice of the cycle, what we would call the M phase. This is where cell division is occurring. All the rest of the cell's life is interphase. So everything else is interphase. It's shown here as orange. We'll just go ahead and call it interphase. Interphase is broken up into three major steps. I don't actually care if you know them or not. But one thing I do want you to know is that this S phase here, this is where replication occurs. If you don't remember what replication is, you probably want to go back and watch the video on nucleic acid structure. But this is where DNA replication happens. The S stands for synthesis. Many cells will actually never divide again. Many cells will remain in permanent interphase for the rest of their life. They'll go into G0, which you can see here, as sort of a permanent step. If you're going to remain in interphase for the rest of your life, you're not going to copy your DNA. You would only copy your DNA, which requires a lot of energy and matter investment, if you are in fact planning on dividing. There are different types of cell division. This diagram breaks them up into three major types. We've got binary fission, which is what we see in prokaryotes. Pretty simple kind of thing. The DNA, which we can see here, is one circular chromosome. Typical of prokaryotes, is going to be copied, and then those chromosomes are going to separate, and then the cell is going to divide, and you can see that happening here. Eukaryotes are going to use two different mechanisms of cell division. We've got mitosis and meiosis. Mitosis is going to be used for growth and repair, and actually for most cells it's going to be used for reproduction as well, what we call asexual reproduction, where one cell is just making a copy of itself. Meiosis is going to be used for sexual reproduction, and really only for sexual reproduction. And so in the rest of this video, we're going to look at these two processes in depth. We're going to leave binary fission alone, and we're going to get into the details of mitosis, and then look at how meiosis takes those and adapts them for the purpose of sexual reproduction. These are the different phases that we would see in mitosis, starting with a cell in interphase. When a cell is going to divide, after it's copied its DNA and it's prepared for division, the steps of mitosis are shown. The steps of mitosis proceed in a clockwise fashion in this diagram. We're going to go first into prophase. What you can see is happening in prophase is you can see that the DNA was very loose in the nucleus, and so now it's being condensed in the nucleus into chromosomes. The chromosomes are just tightly packed pieces of DNA that will be easier to move around the cell because these chromosomes are going to need to navigate throughout the cell if we're going to wind up producing two cells at the end. We're also starting to get the formation of what we would call the mitotic spindle. That's going to be involved in moving the chromosomes around, and this is happening in an animal cell, so it's coming from these regions of the cell called centrioles. The nuclear membrane will begin to disintegrate and disappear in this next step. It will generally be almost entirely disintegrated, and this is what we would call pro-metaphase, which is actually sort of like the end of prophase and the beginning of the next phase of metaphase. We can see that the nuclear membrane is entirely gone now, and our chromosomes are all arranged, and they're beginning to attach to the spindle. The spindle has navigated to the two different poles of the cell. Once we get to metaphase, all of the chromosomes are going to be arranged in the middle of the cell, what we would call the metaphase plate, or the cellular equator, if you want to think about it that way. Each chromosome is attached to the spindle originating from both poles of the cell. In anaphase, the replicated chromosomes are going to separate from each other and be pulled to opposite ends of the cell. And once the chromosomes have navigated to the opposite ends of the cell, they'll begin to decondense. The nuclear membrane will begin to reform. This is what's happening in the last phase of telophase, before the cell splits in half through what's called cytokinesis. And so these are the steps of mitosis. It may be useful to understand what's occurring in terms of number of chromosomes and their relationship to each other. In any eukaryotic sexually reproducing organism, you've got two copies of every chromosome in your body. We call the number of individual chromosomes, we symbolize that as N. And so you have 2N chromosomes in your body. In humans, for instance, it's 23. Prior to mitosis, in S phase, you're going to copy these. And so you're going to wind up, every cell in your body is going to have 2 copies of each of these chromosomes. And so each cell in your body after S phase is going to have 2 copies of the 2N number before the events of mitosis take us back to 2 cells, each with that original 2N number. That N is going to change depending upon the species. So for instance, in fruit flies, N is 4. Yeah. In chimpanzees, N is 24. In us, it's 23. Another thing that's probably useful to understand is the relationship between the different structures of chromosomes that we see. If we look at the chromosomes in prophase, we can see that each one is sort of a double-line structure, as you can see here. That double-line structure represents the replicated copies of each chromosome. They're connected to each other. You can see them here as well, though it's not quite as well defined. Let's see if I can show you how it's separated out. Sort of a line here separating these 2 chromosomes. Generally, this X structure that you would see of a chromosome, what that's representing is 2 copies. And what that is it's 2 chromatids. Sorry, I don't come up with these names, but the 2 chromatids are each identical copies of each other. And we can see them again aligned here in metaphase with the chromatids joined somewhere towards the middle of the chromosome in a region called the centromere. During anaphase, these chromatids are going to separate from each other and be pulled to the opposite ends of the cell, so that the end result of telophase is that each cell winds up with 1 copy of each of the chromosomes that were present in the cell prior to division. And mitosis works for almost all eukaryotic cell division on the planet. Every cell in your body that's being produced, with the exception of the cells that will be involved in sexual reproduction, is being produced through mitosis. It works really, really well and produces genetically identical daughter cells from that parent cell. The issue is that organisms that engage in sexual reproduction need to produce special cells to engage in this process. That's where meiosis comes in. Meiosis is going to be involved in sexual processes, and our friends, these mice, are showing us what happens in sexual reproduction. Anytime we think about sexual reproduction, regardless of the organism in which it occurs, we're going to wind up having cells that are produced that instead of 2N are single N. The word for this is haploid. These haploid cells, which we'll call gametes, are necessary because in sexual reproduction, these 2 haploid cells, the sperm and eggs that are produced, are going to combine with each other in fertilization in order to restore that chromosome number and go back to a 2N or diploid cell. In order for this to happen, there needs to be a couple of differences between the meiotic cell division and that pre-existing mitotic cell division. But the events are still roughly similar. There's 3 major differences that I'll point out. The first difference is that in meiosis, you have 2 rounds of cell division. So starting at the top, the cells will go through prophase 1, metaphase 1, anaphase 1, telophase 1. And then the 2 cells that are produced at the end of telophase 1 will go through another round of prophase, metaphase, anaphase, and telophase. You had an S phase preceding prophase 1, but in between telophase 1 and prophase 2, you do not have an S phase. You do not copy the DNA. This is key because if you copied the DNA, then you would just go back to that 4N number and wind up producing diploid cells at the end. In meiosis, the goal is to produce haploid cells, and so there will be no replication of DNA in between meiosis 1 and meiosis 2. There are 2 other differences between meiosis and mitosis that I want to spotlight that will be really important in understanding genetics. The first is what happens during prophase 1, what's known as crossing over. And crossing over involves what are called the homologous pairs of chromosomes. These are the copies of each chromosome that you get from your parents. You get a copy of each chromosome from your mother and your father. And so these 2 copies of the chromosome we would call the homologous pair. What's happening in prophase 1 is you get physical exchange of genetic material between the homologous pairs. The end result of this process is that every chromosome winds up being a genetically unique combination. This is a really cool thing and kind of a big deal if you think about it. When you pass on your chromosomes to your children, if you decide to have children down the road, you will not be passing on the chromosomes that you got from your mom or from your dad. Instead, you'll be passing on unique chromosomes, where each one is a unique combination of the homologous pairs of chromosomes that you got from your parents. This leads to a massive increase in the amount of variation present in sexually reproducing organisms, and it also means that every sexually reproducing organism is a genetically unique combination of the genetic material that they inherited from their parents. The other major difference is what happens during metaphase 1. In metaphase 1 of meiosis, the homologous pairs, which have just engaged in crossing over, wind up lining up at the metaphase plate, still attached to each other. This is different than what it looked like in mitosis, and you're welcome to go back and take a look at that if you want to see the difference. But this leads to what's known as independent assortment of homologous pairs during metaphase 1, which is going to be important later on when we talk about Mendelian genetics. So keep it in the back of your mind. It's probably a good idea to compare mitosis and meiosis here at the end of this video, and so this chart helps us do this. The purpose of mitosis is for growth, repair, and asexual reproduction, whereas the purpose of meiosis is to produce gametes for the purpose of sexual reproduction. In mitosis, there's only one round of cell division, whereas in meiosis, there are 2 rounds of cell division with no replication in between. And the other major difference is the products that we get. At the end of mitosis, we get 2 diploid, or 2N, daughter cells that are genetically identical. They're clones of each other. Whereas at the end of meiosis, we're going to get 4 haploid gametes. In other words, they're N, and they're each going to be genetically unique. If we want to think about the overall equations for these processes using this idea of the haploid number, or N, in mitosis, we're starting with 2N, and then after S phase, but before mitosis, we've produced inside of that cell 2 copies of that 2N. This is then going to give rise at the end of mitosis to 2 cells that are each 2N. In meiosis, we're starting with 2N, and prior to undergoing meiosis, we're going to copy the DNA as well. So we're going to wind up with 2 copies of 2N. After meiosis 1, we'll have 2 cells that are produced that are each 2N. But then, because we don't copy the DNA again, at the end of meiosis 2, we'll have 4 cells each with the N number. This might be helpful to get your head around what's happening with the genetic material during these processes. As we move into check your learning here at the end of this video, make sure you can do the following things. Make sure you can explain how and why cells divide and the different purposes of the different modes of cell division. And then make sure you can describe, compare, and contrast the processes of mitosis and meiosis. If you can do those things, you're doing great. If not, that's okay too. Take a moment and pause the video and write down any questions that you have so that you can get the answers that you need. Thanks again for watching. I really appreciate it. Have a great day.